I guess the more I tried to make slides for this talk and the more I've listened to every one of you talk today, the more I've realized that I'm someone who focuses on kind of the parenthetical subplot of things as opposed to focusing on, I guess, the main why question. So I apologize if this talk is a little bit how heavy, but I hope that towards the end we can start thinking about the whys and I guess spur the discussion straight after this. So this is, of course, a talk about aging, so we have to include this obligatory slide about the hallmarks. But the reason I've actually included this here is that in this particular review, when they talk about loss of proteostasis, they highlight it with this figure, and it really illustrates the point that I want to be talking about perfectly well, which is the idea that you have some sort of proteotoxic stress, which leads to protein misfolding, and then it is up to the cellular quality control machinery to decide what to do with that misfolded, unfolded protein, whether it could be refolded, whether it needs to be cleared, or whether it needs to be sequestered somewhere. And so the first part of this talk, actually most of this talk, I'm going to focus on some of the molecular mechanisms by which we understand this triage process to take place. And so I kind of go back to this, I guess, relatively old figure, 1997 figure that I found, which frankly could have been published today, and the details are almost the same in terms of what we think about in terms of triage. And so you have a protein which is present in multiple different folding states, and the QC machinery needs to find some way of finding all of these states and then distinguishing which ones of these are suitable for folding, which ones aren't and instead need to be degraded, and which of one of those really can't do either and instead need to be sequestered away from the cellular milieu. A couple of years later, the same authors again added the idea that in eukaryotic cells, you also have ubiquitination, where a misfolded protein can have a ubiquitin chain attached. And once that chain becomes long enough, and we usually think usually around 4 ubiquitin molecules is enough, it gets recognized and degraded by the 26S proteasome. And this is really where we are right now. So even though our molecular understanding of these options have improved over the last, I guess, 20 or so years, the basic mechanisms by which this decision takes place is still really, I guess, relatively unresolved. And so thanks to Georgios's beautiful talk about chaperones, I can get straight into the meat of the problem. And so this is the idea that an unfolded protein is going to be bound by a chaperone, and the chaperone uses a series of ATP binding, hydrolysis, and release cycles to help the unfolded protein attain a more folded structure. And then this is bound again by a chaperone and then helps it fold again, and this becomes more folded. And so eventually, through a series of binding and release events, the protein, if all goes well, will reach its native state. And the important thing to remember here is that even though this reaction, if things go well, tends towards the native state, it's still stochastic at an individual molecular level. So it's really unpredictable whether an individual binding and release event will actually lead the protein to becoming closer to the native. And so this is again what George just talked about in terms of the energy folding landscape. And so while the native state does represent the, I guess, desired trajectory, there are several kinetic traps that the protein can get stuck in, and what could be even worse is it gets stuck in traps that are actually at a lower energy state than the native protein. And so the cell needs to have a way of avoiding this scenario. And so this is really one of the ideas for the molecular basis for triage. And so we have this idea where you have a native polypeptide chain that's released from the ribosome, and it gets engaged by a chaperone. In this case, it's for a tail-anchored protein, but the identity is unimportant for now. And so this chaperone-bound nascent chain has one of two possible fates. The first is that it is bound by a cofactor or kind of a trafficking agent that takes it away to do its physiological role. Now, this transfer is fast and it's irreversible. However, it's also very selective. The protein needs to be in a native or near native state for it to be recognized and transferred onto the downstream effector. If it isn't, then it remains bound to the chaperone. And the longer it remains bound to the chaperone, the more chance there is for an alternative transfer to take place. And this is to the degradation machinery. Now this, in comparison with this first reaction, is much slower, but it's much more tolerant and promiscuous. So regardless of how many non-native states this protein may sample when it's on the chaperone, it still has an equal chance of being transferred to this secondary pathway. And so basically, the longer the residence time of the protein on the chaperone is without it reaching its native state, the more chance there is that a degradative transfer will instead take place. And this is really something that exists to avoid the cell expending useless kind of time and energy in trying to fold proteins that are terminally misfolded and really are never going to reach their native conformation. In order for that statement to be true, there needs to be off-ramps at both the unfolded and the folded side of this kind of schematic. And in this case, I showed you it was achieved by competition between two different trafficking factors. And this seems to hold true in general. So going back to our beloved HSP70, HSP90 machinery, pro-folding co-chaperones or pro-degradation co-chaperones, in this case a ubiquitin ligase, compete for the same site on HSP70 and HSP90. And so in this case, you have the triage decision between folding and clearance dictated by direct competition between different co-chaperones. This isn't just true for the folding versus degradation decision. It's also true further down the road. So in this case, when we look at different co-chaperones, so the bag proteins, there are one set of bag isoforms, the bag ones, which if they bind each of protein 70, they send a protein to the proteasome. If instead HSP70 binds bag 3, that client protein is sent to the autophagy lysosome machinery. And I guess the reason I've decided to highlight this is because there seems to be a shift during aging or replicative senescence from the bag 1 mediated proteasomal targeting to the bag 3 mediated lysosomal targeting. And so we have this kind of idea where in kind of young, fit, healthy cells, the bag 1 mediated proteasomal targeting route is sufficient to keep the misfolded ubiquitinated protein load in check. During aging, this becomes imbalanced due to the accumulation of protein aggregates until eventually it triggers a transcriptional switch that leads in upregulation of bag 3. And so now proteins that in young cells were targeted to the proteasome via bag 1 were instead taken over by the bag 3 mediated pathway. And this is a way of restoring imbalanced proteostasis. And the kind of sensor for this switch would be an overload of the proteasome, which we know declines during aging. And so the idea is that instead of keeping misfolded proteins sent to the proteasome, it doesn't matter how much bag 1 you have if the bottleneck is at the endpoint of the pathway. So instead, the cell chooses to get around this bottleneck and activate a more promiscuous pathway that bypasses here and now leads to degradation of everything that would have been blocked through the lysosome. The problem here, of course, is that we still don't really have a good understanding of why this switch occurs. Again, I'm not very good at the whys. So you could imagine, I guess, two different scenarios where you have protein aggregates that accumulate during aging, and these we know are poor substrates for proteasomal activity. So this leads to the proteasomal overload and hence the bag 1 or bag 3 switch. Conversely, you can imagine that this actually happens the other way around, where you have an overload of the proteasome. This means that more and more misfolded proteins don't get degraded, they accumulate in aggregates, and this is what drives the switch. So this is kind of getting to the problem we often have with any kind of aging-related study. It's really difficult to attribute first cause when there are so many things happening at the same time. So it also kind of illustrates a trap I fall into very often, which as a chaperone biologist, I think about triage purely from a chaperone-centric perspective. And of course, this is missing the very important point that the actual qualities of the substrates that are being forwarded themselves really are going to have a big part to play on how these two factors affect triage. And so really quickly, I'm going to go through a couple of these, I guess what I would call canonical rules governing clearance. And so one of them, I guess one of the first things I learned when I came into this field was that the idea that monomers go to the proteasome, whereas oligomers and aggregates go to the lysosome. And this is relatively intuitive when you think about how the proteasome works, because it needs to thread a misfolded protein kind of linearly through a narrow channel in order for it to be chopped up into peptides. If you have an oligomer or an aggregate, well, one, it's kind of difficult to unfold this and unthread it. However, even if it wasn't difficult, it's just really time consuming. So the idea that you want to get rid of an aggregate by threading through each polypeptide within the aggregate one at a time is just really, time consuming. It'd be much quicker to send the whole aggregate over to the lysosome. So I believe, actually, David Smith's going to be talking about this in a couple of days' time, and I stole this figure from one of his papers, so I'm not going to talk about that anymore. Instead, I thought I would talk about how some of the rules I've already mentioned in the first half could relate to the decision to send proteins to the proteasome versus lysosome based on the oligomerization state. So what I'm going to use to illustrate this is really a couple of beautiful papers from the late Stefan Jentsch's lab, where they looked at two ubiquitin binding receptors. So these are parts of the proteostasis machinery, but they bind ubiquitinated proteins. And so this is in yeast. And so the first of these is called DSK2. In mammals, it's often called ubiquilin. And so it binds a polyubiquitinated protein and shuttles it to the proteasome to be degraded. The second ubiquitin receptor, Q5, also binds ubiquitinated proteins, but instead of sending it to the proteasome, it takes it to autophagy. And so when you look at the relative affinities of these two receptors for a polyubiquitin chain, DSK2 has a much, much higher affinity. So if everything else is the same, a ubiquitinated protein will be preferentially bound by DSK2 and sent to the proteasome. Now, the reason why Q5 has any role to play at all is that it has the ability to form higher order oligomers, whereas DSK2 does not. And so you can now imagine a scenario where if the ubiquitinated protein persists in the cell for long enough, either because the proteasome isn't working and is overloaded, or because the protein itself that's ubiquitinated has the propensity to aggregate, now you can have these higher order oligomers forming on the ubiquitinated aggregate. And instead of having efficient proteasomal degradation, you now see a shift kind of in avidity to preferentially bind Q5 oligomers and send it to autophagy. So I think this is just a really elegant example of how the examples I've given earlier, where co-chaperone competition and residence time can really have a role to play even much later in triage than the folding versus degradation decision. The second point I kind of wanted to touch on is based on this second textbook rule where short-lived proteins supposedly go to the proteasome, long-lived proteins go to the lysosome. Now, aside from the fact that this definitely represents trends rather than rules, there's a ton of exceptions in both directions, and it's also very context-specific. So I can go into this further later if anyone's interested. But it also really applies to the mature form of the protein. So I don't think anyone's arguing that an actin monomer, if it becomes misfolded off the ribosome, it's going to be preferentially sent to the lysosome as opposed to be, QC'd by proteasomal pathways. Nor, I don't think, is anyone trying to say that transcription factors and cell cycle proteins that happen to form aggregates will still be preferentially degraded by the proteasome as opposed to going to the lysosome. So when we're talking about this specific rule, we're very much talking about the mature form of the protein, the mature half-life. And this is where we're really getting to, well, in the last 2 minutes, the headline of the talk, which is trying to think a little bit more about the whys. And for this, we need to talk about regulated versus quality control degradation. And so, as I said, when we're talking about this rule, we're mostly talking about the mature half-life of the protein. And when we understand turnover of the long-lived proteins, we're generally talking about turnover due to damage, due to things like oxidation, carbonylation, various other types of damage. When we're talking about the same for short-lived proteins, we're generally talking about degradation because the signal needs to be dampened. So all of these are very tightly regulated at both the transcriptional and the translation level, but also at the protein level, because it needs, just as important as turning on a signal, let's say for an example, the signaling protein, also need to turn off the signal, otherwise various bad things are going to happen. So I'm just going to illustrate this with 1 or 2 points, starting with the cell cycle, which is that really beautiful work from Misha Rapa's lab, as well as a ton of others. The idea that here in the cell cycle, you have dozens of different transcription factors, cyclins, kinases, phosphatases, DUBs, E3s, all of which need to be active at the right time, and just as importantly, deactivated at the right time in order for the cell to progress through the cell cycle. And we understand relatively well how this is done at the level of ubiquitination and degradation. And this is done by the anaphase promoting complex, which is a massive multi-subunit ubiquitin ligase. And it really employs each of the mechanisms I've talked about earlier to achieve this ordering of processivity. So the actual initial binding of substrates to APCC is controlled by relative rates of 2 different cofactors of this enzyme. So the cell can control the actual synthesis of these 2 co-chaperones, so it's UC20 versus CDH1, at different times during the cell cycle. And so it can switch by cofactor competition which of the substrates are going to be bound by APC first and which are going to be bound, let's say, in the second wave. An additional level of complexity is provided by the fact that some substrates have really slow off rates on APC. So a single binding event of substrate with the ubiquitin ligase will lead to enough ubiquitination for that protein to be directly sent to the proteasome. However, there are other substrates which are much, I guess, more meandering on their way to being polyubiquitinated, with a series of binding and release events required. There's also deubiquitinases involved in parts of this pathway. So really, again, you can now see that how residence time on the E3 ligase is affecting the ordering of processivity. I didn't really have time to talk about this, but ubiquitin chain complexity also plays a role. I'll be happy to go into that at the end. But this was an example of the cell cycle. It's also true for things like transcription, where in this case for a GAL4 transcription factor, it's activated by a monoubiquitination event, and it remains active for as long as it takes for this monoubiquitin to become a tetraubiquitin chain, at which point it becomes degraded and the signal turns off. So this is intrinsically built into the system for activation. Similar thing with apoptosis now, where in this case you have a single ubiquitin ligase that activates the caspase, and then second ubiquitin ligase that binds only after the activation has taken place to now trigger degradation. And just finally, to wrap up this idea that ubiquitin chain elongation plays an important role as a timer, all of those examples were in kind of a very regulated degradation. It's also true for quality control, because at least in eukaryotes, with the 26S proteasome, it has a built-in ubiquitin ligase deubiquitinase pair. And so by tuning the relative levels of these 2 proteins at the proteasome, the cell can change the residence time of ubiquitinated substrates. For example, if you increase the levels of Hal5 at the proteasome, any proteins that are bound to the proteasome will get chain elongation, and so have basically an increased chance of being productively engaged and degraded by the proteasome, and vice versa if you increase double levels. This is kind of what I wanted to finish with. This idea that successive ubiquitylation events are really a conserved part of triage and degradation, where within every subsequent ubiquitylation event leads to more of a chance of the substrate being engaged and committed to degradation. And hopefully that will be quite reminiscent of what I talked about at the very beginning, which is this binding and release by chaperones being kind of, I guess, increasing the chances of proteins reaching their native state. And so these are kind of two almost opposing models, where here from left to right you're going to folding, here from left to right you're going to degradation. But I hope I've kind of touched on the fact that a lot of the concepts that are used at the molecular level to make these decisions are conserved. And so our lab and quite a lot of other groups in this field, I think, are now trying to reconcile and kind of integrate these two different ways of approaching the protein triage world. And hopefully this is something that we're going to be able to give a more coherent model for how triage really works at a molecular level. So yeah, with that, I'd like to thank you, and everyone kind of knows who they are. One thing I'd like to say is I don't have a group photo because none of our group have actually been in the same room yet. Hopefully post-COVID, if I get invited again, there'll be a nice photo of us outside somewhere. But yeah, so I'd like to thank everyone, and I'll of course take questions. Thank you.